I am Bjarke Fenstra. I'm a senior researcher and group leader at Statens Serum Institute in Copenhagen, Denmark. I'll talk about our new study on febrile seizures. It's a frightening experience for parents to witness their child have a febrile seizures episode. Most febrile seizures are fortunately benign and don't recur, but in some cases febrile seizures are followed by epilepsy in a child. About 4% of children in Denmark experience febrile seizures. It corresponds to about one child in every school class. So our goal with this study was to understand the genetic background of febrile seizures in greater detail. For the study, we conducted this as a genome-wide association study, including more than 7,000 patients with febrile seizures and more than 80,000 controls. Most of the samples were taken from the Danish National Biobank, but others came from other groups in Denmark and collaborators in Australia. In our study, we discovered 7 new loci associated with febrile seizures, and we confirmed 4 previously described loci. I will talk about 2 of those loci a little bit in greater detail. One locus is related to fever response. It's near the gene PTGER3, which encodes a receptor for prostaglandin E2. This molecule is a central player in the fever response. And in mice, it has been shown that if the gene PTGER3 encoding the receptor for prostaglandin E2 is knocked out, the mice are not able to generate a fever response, even if they are provoked with triggers that usually provoke fever, such as lipopolysaccharides. We found that the SNP that was associated with febrile seizures is also an expression QTL for this gene in various brain regions. And the direction of effect is according to the expectation that the risk allele for febrile seizures corresponds to increased expression of the gene in brain tissue. The other locus that I will talk about is harboring the gene GABRG2, encoding a gamma-2 subunit of the GABA-A receptor. And GABA is the major inhibitory neurotransmitter in the central nervous system. It has been shown in previous studies that rare mutations in this gene altering the protein are associated with epilepsies in families in a dominant fashion. And these syndromes often also include febrile seizures as part of the presentation. The new thing about our study is that common variants in the gene are shown to be associated with the much milder outcome of febrile seizures. More generally, we also found connections between febrile seizures genetics and epilepsy genetics. We found that there was a positive genetic correlation between our results and results from a previous genome-wide association of epilepsy. We also conducted polygenic risk score analysis. Here we found that polygenic scores for febrile seizures were associated within patients with a higher number of total hospitalizations with febrile seizures and with an earlier age at the first admission to hospital with a febrile seizures diagnosis. Finally, we compared our results in the so-called iPsych study of various neuropsychiatric diseases. And here we found that febrile seizures genetic polygenic risk scores were not larger in disease groups of the iPsych study compared to a population representative sample. In conclusion, this is the largest genome-wide association study of febrile seizures, and it illustrates that genetic loci related to fever response as well as to communication between nerve cells and neuronal excitability play an important role in the genetics of the disease. It also underlines connections with epilepsy, which we hope to be able to explore more in future research. Thank you for your attention. You can read additional details in our article.